Okay. Nicely done.
That's enough?
Yes, that's perfect. Thank you so
Now I'm
much.
starting to filter.
So that little, this is the first one, I guess the hole's just over the top here.
I believe so. I don't, yeah, it's just over that.
Not sure where the mark that from.
Yeah, I don't know where that is.
The marker's in the port side and the aft.
Yeah, no, we're talking about on this,
Oh.
this view.
Like this is the waypoint, is this not?
Yeah, this is it. We're there. There's no X anymore.
There's the milk crate.
That's what we're picking up.
Okay.
When we recovered the temperature logger on the last dive,
Yes.
marker X was attached to it. So we're going to be putting the logger back down after we get Jeff's osmosis in. But I think first we're doing the super, right?
Yeah, I think the plan is to pull that out of there, pull the intake out, maybe set it aside,
Yeah.
so that you have a clean hole, and then.
Do your DO.
No, I think you want to do that last.
Okay.
So what we'll do is,
Take a print out, please.
what is this?
Oh, that's fine. Yeah, that's fine. Thank you. Okay. I'll let you direct from here, Jeff.
Okay.
And
Oh yeah.
What a great view.
just keep going, going.
Yeah. It's a really big anemone.
So what we'll do first is, you can try to reach it. Yeah, I mean, because we didn't place it there.
Are we putting the phone with the crate all the way, or are we trying to grab the line and let it go on?
Yeah, so we're gonna, no, yeah, grab the, what you wanna do is grab that ledge and what I would do first is if you can reach down, try to grab the web piece. If you can't, we'll use the hook. But then what I would do is I would swim just a little out like this and come up like this so that you don't get caught in the hole.
There's a little 2-pound dive weight holding it down at the intake. So in this milk crate that we're about to move, just to get it out of the way before we do other things, there's 2 spools of Teflon tubing and 2 pumps, and the pumps are osmotic pumps. There's a membrane that separates saturated salt on one side and fresh water on the other side. And that gradient, salt gradient, drives flow across the membrane. And that's the pumps are attached to the small bore Teflon tubing that then pulls in the water through the tubing and through the intake at in the bottom of the hole. And then when we get these back, we cut up the Teflon tubing into 1 meter strips, get the water out, and now we have a time series of water that came out of this orifice. And with that, we can analyze them and not only tell the composition, but see how it varied over the past week that these have been out here. Others have been out here for 6 months.
Try to stop there. Try to straighten out your lower arm. Just straighten out that joint. Arm up. Yeah. Slide up top. Grab it. Give you some feeling sit.
Yeah, nice job.
Slide up. Yeah, so what you want to do is pick it up a little bit.
Jeff, if you zoom real in there, can you see water?
Um,
I'm just going to keep the camera here. 'Cause I
okay,
think it's,
yeah, yep.
Okay, so, okay. So just gently lift up and then I'll fly it up and I'll make sure you're clear. Yeah, it's free here, dude. That's it.
So at the intake, there's a little float, and on that float is a dive weight or attached to it, and the intake for the osmo sampler as well as a temperature logger. So you get an idea of what the temperature is too of the water.
If you want, what you can do is maybe I can fly up and I'll go off to the side. Just drop it up. It's not so I could shove it up. Sure it's on that. Yeah.
On the side,
What we'll do is I'm going to fly over to the edge of the ridge and we'll just set it down gently.
it's a phase.
So the reason why we're pulling this one out first is because we're going to put a long snorkel in to collect water next, and we just didn't want to have a whole bunch of lines in one place. So this is just going to get, we're going to put this on the seafloor for until the water sampling operations are done. And then we'll pick this up and head off and grab another animal and come on up.
This one coming through.
Yeah. So I'll just lower this part down. Alright.
Wonderful.
Jared's asking if one of you all could stay free.
All right, so now we're heading back to that hole where there's water coming out of it. And we weren't able to get a really good sample last time just because it's so far down. So now the intake has been modified to be able to reach down there, and we'll use the pumps to pump out some of the water that's coming out. And hopefully it'll tell us a good story. One of the questions about the white layer that you see every now and then, there is a carbonate crust around here, and I don't know, 2 hours ago or something like that, we cored some of that material where it was a lot thicker than just the little bits that you're seeing in this section.
So, which one do you want to start with first?
Well, whichever one you want to do first.
Probably, what do you think? I'm thinking, because that one's got a floaty line. Maybe we do that last.
Yeah.
So I'll say first.
Sounds good.
Okay.
You don't have temperature as a guide this time?
Okay. So we're just gonna get it as far in as we can?
Yeah, and Jeff, it's sort of the right side of that hole, right?
Yeah.
What's that?
Yeah, so we're gonna want to try to get it over here on this in this part of the crack. Wait. So that one's the.
Yeah, we're just getting untangled.
Oh, okay.
It's not a problem. As long as that rail's on it, it's fine.
So the PVC cap that's attached to this intake is going to act as a funnel to hopefully help us get the most pristine fluid.
Jeff, the thing we just pulled out,
Yep.
did it also have a temperature logger?
Yes.
Okay.
And the DO has a temperature logger in it too.
Oh, that's right.
So we got a couple of them.
So you can drop it there.
We modified the intake with like, it's the super sampler, but it's just a different intake to try to get down in the hole. Yep, it's still super. Yep.
So the hose is attached by sort of weak zip ties on the basket just to keep everything manageable while we're driving around and doing other operations. And now that we're here, we're loosening them up and lowering the cap into the hole.
Where is this? 50. So should we move it to the right, Jeff?
You know,
Let it.
That went down the hole pretty good.
It
Yeah.
sure did. I don't want to suck up all that sediment.
We can, I guess, come under it and just try to.
Well, I sort of,
What are you thinking?
I'm not sure since I've never deployed anything down the hole. What do you think, Jeff?
well, why don't we wait a couple minutes before you start pumping?
Do you think we should try dragging it all, or do you think the whole hole is?
May kick up more sediment down
Yeah.
there. I don't want to drop that rock down there.
Okay.
That camera just always walks up when it looks straight down, but I gotta get
Okay.
it here.
Oh, yeah. Yeah, no, that looks.
Like it looks like it's
Good.
actually hanging over this
Yep.
one
Another
rock.
ledge, right?
Yeah, so you're probably looking all right.
Yeah. I don't see any shimmering.
Did we ever?
Yeah. minutes and then don't start with your filters.
No, we do the bottles first, then the filters.
Yeah.
I thought I saw it, but I couldn't tell. It was just like flecks of something. You think that's flow?
Well, I can't tell if that's bottom currents just, or if it's hydrothermal.
Either.
I mean, I don't see any, yeah, I don't know.
Alright, I'm gonna start my pump, see. Make sure it's flushing through. That looks shimmery.
Yeah, but I think that's just bottom currents.
Yeah?
But it's nice to see the crabs.
Right.
Well, we did see a temperature change in the hole when we.
No, we did.
Yeah. Are you gonna fill all the bottles?
Not all of them. I think I'm pretty short on bottom time, it seems. But I'm gonna start bottle 8. Right now. Bottle 8.
So right now we've got the intake down that hole. Previous trips, we've seen water coming out of the hole, and it's a little difficult to see any sort of water coming out of that hole. That's hydrothermal, not just bottom water flowing by. This is just right above the hole and probably connected to it. Right now I'm just looking around here to see if there's any other clear signs of flow.
Yeah, I got back. Isn't this gorgeous there?
Where is that, Jeff?
Right above the hole. So it's probably connected to the hole itself.
Right. Can you zoom down on the right side of the hole? Because I feel like maybe it's a little. Oh. I thought that whole hole is just full of it.
Yeah. I don't see any coming up this crack.
Maybe you do. I don't know. It's so hard to tell.
Looks like there's a little sediment inside this guy.
Is all the tubing that we have in the basket out?
Yes.
Yep.
That was like 25 feet,
Seriously, that much?
I think. 'Cause I brought 2 lengths of that tubing. They're each 25 feet. I think we cut the original intake a bit. Done the sound flexing. Did we get it?
Right now what we're doing is we're sitting here pumping some water out from the hole down below where we have seen good shimmering water come out earlier. And so right now we're just pumping it away, and while we're doing that, we thought we'd scan over and see what sort of critters were on the seafloor and see if there'd be anything fun and dynamic happening in this little underwater world.
We're actually moving.
I have this memory of like the drawer under my bed slamming open, various things. Like I heard a lot of slamming doors last night.
Well, it could be that stuff that wasn't latched before, all of a sudden shit's moving. I sat there laying in bed thinking, I think I have a bucket of acid in the hood. I should like go pour that out.
Yes. Okay. I'm gonna go grab breakfast, but then I'll start
Okay.
washing bottles and etc.
Yeah. Could you put a lunch plate in this iPhone? I mean, but I'm gonna be doing this for another hour, so. Okay.
Although, I mean, I'm also happy to swap out for you that much because if I go eat breakfast, come down
How's the pumping going?
It's going really good.
Good.
Yeah. Pump, pump, pump.
Yeah.
A little wheelie thing moving. So, okay. That's what happens when you're in October.
Yeah, I'll be right back.
Okay. Okay.
I mean, we got another half hour sitting here?
So let's see, is the only other operation getting this out, putting the DO probe in,
Waiting 10
minutes, taking it out, stowing everything and leaving?
I think there might be picking up a critter at the very end.
Do we still need to pick up a critter?
There was discussion about getting a Brittle star, but
Okay,
how are things going?
they're good. We have 25 feet of tubing down the hole.
Yeah,
Well,
look at that.
not all down the hole, but
And is the temperature?
we don't have a temperature sensor on it.
Oh, okay.
Yeah.
Okay.
Need to leave bottom at 12:15. Is that right?
Ish. Yeah, so I think if we, and what do we still need to get the milk crate on board?
I'm stopping that sample.
We got it off to the side right now.
And we haven't deployed the marker,
Yeah.
and we need to deploy the dissolved oxygen probe.
Yeah, he wants it to sit there for like 10 minutes.
Yep. So,
I'll try to be
does
done
11:30 work for you?
for fluid sampling. I think I'll try to be done by 11:15.
Okay.
I'm starting level 10,
Bottle temperature for the last one?
but we don't have temperature.
Oh, gotcha. Bottle 10.
Yeah, that works.
It's fine.
Can I see your? Just look at this.
Are we okay to just drop the marker right after the leak and just run this like on the rigid event?
Yeah, kind of like where that anemone is or whatever. That's anywhere here is fine. Just long as it's in the hole.
Okay. Maybe do you want it so if we put it up on the ridge here so it's easy to see from far away?
Sure.
Okay.
Yeah, so once you've done whatever order you want, like the osmo sampler and then with the marker and then we need to drop chips, dissolve oxygen probe.
Okay.
I figured once you might as well do it your last operation.
So were you talking about doing a brittle star? Because if we
There was a request, it's not essential.
Okay. I was gonna say, we're not sure, this is floating line, so
Yeah.
once we drop that and recover it, it may, that we have to hold the line, just manage it.
Oh, so we didn't put any weights on it?
We do have some weights on it, but
Okay.
we're not sure how we're gonna stuff all the stuff out on the
Yep.
porch, so. I guess we're already here though. Yeah.
Yeah.
Figure it out.
Yep, we'll figure it out. Like if we get done with those apps, then we still have a couple minutes.
Yeah, these are more important anyways.
Yep.
Okay. Cool.
Morning, good evening.
Whatever. Yeah.
Good day?
You guys are playing video games.
Yeah, indeed.
Amazing.
Life is a video game, though. Especially for these guys.
I'm sure.
They've got steady, very steady figures under pressure.
Yes. So do you think you can get your arm under there, just kind of like,
Okay.
just get the back rope, and I'll try to like.
Looks like
just want to catch that piece down there.
we're able to get some myosine mud earlier.
I don't even know if it's scarpins.
Mhm.
Yeah, we didn't know they were gonna be here. We're hoping to go back to Fuente.
Maybe you can
very, very last night. Sample it, but we got a bunch of it.
Just so you can see it.
Yeah, there are 3 different levels.
So
Nice.
that's, hopefully we start at 5 million years, go down to close to 20.
Very cool.
Yeah. microbiome for
Is this
that.
253, Oscar?
The one that just started?
Yeah.
254.
254,
253 was the
sorry.
one that ended.
Thank you.
I was like looking for opportunity.
She's just seeing if you're on your toes.
I was off by an hour, so I'm pleased to know it's 10 a.m.
How many more do we have?
I want to tell you. You can leave if you want. I can wax poetic about stuff. Or are you on watch, Gus? Oh, Gus.
Oh, Gus is here.
Gus can
Perfect.
definitely wax
Oh,
poetic.
Yeah.
Am I taking over you? Yeah.
Sure.
Okay.
I'll be back though.
Can I get some shut-eye?
No,
No?
not
Stay
yet.
Okay.
See how we can get all this stuff out of here.
Mhm. Have we got marker X?
Yeah.
Yes.
Oh yeah, we haven't deployed marker X either.
Yeah, that's the very last.
We've
I
got your
will try to
osmos.
be done by 11, which gives us a full hour and 15 minutes for all that stuff.
For the DO.
Well, pulling this out, which
Yeah.
might be a little tricky. Putting your DO in,
Yeah.
getting the marker out, and getting the milk crate stowed.
Okay.
So it might be like 11:10. So we're supposed to be off bottom at 12:15. We got us to the bottom early, so
Well, they got us to the bottom early.
we were ready. The ship got us.
How long have we been slurping fluids, Joe?
A half hour.
Yeah, let you get in here.
All right.
Have the Spanish. Oh yeah, thanks for all the prompts.
Anytime. I could have done more. It's just the stream.
Oh, perfect. Perfect. Does it go down often?
No, we just had a drop out. But it was like super momentarily, but the settings were a little different.
50.
Surprised it doesn't happen more frequently.
You know, it actually used to, especially, I mean, I wasn't on Falkor, but apparently it would drop out like really experiments. Pretty lucky. How you doing, Gus?
Gus is
morning.
ready.
Gus is ready. Hold on. Let me unmute my mic so you can hear me too.
All right, I'll be watching.
Yeah.
I'll come back.
Yeah, we're good.
Good morning, everyone. My name is Gustavo Ramirez. I'm an assistant professor at the California State University in Los Angeles, and I've just taken over the narrator spot from Professor Jeffrey Wheat from the University of Alaska, Fairbanks. Today we are returning to another seamount that we visited before on this cruise. The name of the seamount is Tengosed. That means I am thirsty in Spanish, and that is a very germane name for some of the phenomena, or I guess it's based on some of the phenomena that are happening here. So if you've been listening to our live streaming, you've probably heard a lot of us talk about the fact that deep water is circulating through the ocean crust, and that these outcrops and hills and seamounts, all made out of basaltic rock, serve as conduits for this deep water crustal circulation. This particular edifice here, Tengosed Mountain, it's quite a big one, and it appears to be a net intake place for bottom water, bottom cold water, that then infiltrates the subsea floor crust and moves towards another place that's called Dorado Hill. Once the water that's entered Tengosed begins to exit at Dorado Hill, it's got drastically different properties. It's definitely warmer, so this is considered hydrothermal venting. It's got a different chemical signature, and most importantly for what we're actually doing here now, it's got a different microbiological signature too. So we are currently sampling as much hydrothermal venting fluids as we can, and again, this hydrothermal venting fluid, it's not just telling us what's happening in the fluid, but it's really a window as to what's happening in the entire flow path of the fluid, tens to hundreds of meters beneath the site of venting. So that's really what we're after here. I am joined in the room by Dr. Julie Huber, who's a senior research scientist at the Woods Hole Oceanographic Institute, and undoubtedly a world leader in terms of sampling hydrothermal fluids at many different sites all over the world. Her, along with Amanda Sutherland, another Woods Hole Oceanographic institution person, I believe she's an engineer there, have put together this very sophisticated filtering apparatus that allows us to basically, in a very clean way, take up as much uncontaminated fluid from even a small crevice where there's hydrothermal fluids venting, and we're able to bottle that fluid basically by displacing sterile fluids into many bottles, and by moving this manifold, it's also able to do a couple of other functions, one of them being fixing something called RNA in place. So when we extract DNA from these fluids, we get the total genetic potential based on the genomes, or the blueprints for life, that all of these thousands of species and hundreds of thousands and millions of microorganisms, sometimes per drop of seawater collected, have. Again, but just because we see all of the genetic potential of these microbes, we're not sure what genes are actually being expressed. So each gene ultimately codes for a function, but we don't know what their functions in their natural environment actually are. So we ultimately, I guess long story short, we want to know what they're doing, and there's no way to know what they're doing unless we sample something called a message, or an RNA. So information in biology, I guess, moves from long-term storage in DNA to an active message in something called an mRNA, or messenger RNA. And then this messenger RNA ultimately goes into a protein factory. We might remember that from high school biology, a protein factory called the ribosome. And that's where that message, that's again information that's long-term stored in DNA, that's where that message becomes a protein. And then that protein interacts with the world. So yeah, long story short, what Julie's after here is basically high volume filtering of fluids right at the venting site. So very uncontaminated fluids, and Julie's got this machine that allows us to fix the transcripts, the RNA, in place, so we don't disturb the function. This is a very, again, sophisticated molecular experiment that will allow us to preserve this very short-lived messages of RNA that tell us exactly what the activities of these microbes are in their natural habitat, which is an extremely difficult thing to do.
Can you tell us a little bit about like what the microbes look like say you've sampled a rock, you've cut it open. What does that look like? I
Oh,
just
yeah,
wanna
yeah.
give the viewers a sense.
Yeah, absolutely. Yeah. So, if we were to look at these microbial communities under a microscope, yeah, like the individual cells don't look terribly exciting. They're just basically two shapes. They can be little rods, or they can be little spheres. For the most part, sometimes the rods are a little bent in the middle. Sometimes the spheres come in pairs. But in terms of the appearance of microbes, they all more or less look the same. And the reason for that is, of course, physics. If an organism, like a unicellular organism, like a bacteria or an archaea, have committed to remaining very small, they have they need to guarantee that their membranes have as much surface area as possible. And the best way to maximize surface area for bringing in food and for breathing out exudates, the best way to do that is to stick to these two shapes. So that's why microbes under a microscope are not terribly exciting, and unfortunately that was most of microbiology since the golden days of Louis Pasteur. So they weren't terribly exciting in that sense. However, microbes have been on Earth for much longer than animals have been, and this 3 billion years plus of evolution has led to remarkable achievements by microbes. These achievements, however, are not in terms of their form. So a lot of times when we think of evolution, we think of so-called advanced or we think of like higher animals in evolution because they tend to be more morphologically complex or something. But that's never going to be the case with microbes. They look the same. They look the same 3.5 billion years ago. They will look the same probably 2 billion years into the future also. But what's been remarkable about them, what they've been up to, is evolving truly novel metabolisms. And these microbes are able to do incredible things that humans that we could really never imagine. In terms of our metabolism, humans, right? We are heterotrophs, so we're stuck needing to eat at least 3 times a day, I hope. And every time that we eat, right, we ingest organic molecules that are loaded with electrons, and those electrons need to go somewhere. We provide a final resting place for those electrons by also needing to breathe. So there's eating to get organics with a lot of electrons, and then breathing air to access that 18% of the air that's oxygen. That's a wonderful oxidant. It's basically all that we can do. So heterotrophs, like most animals, including humans, we're kind of a one-trick pony in terms of our metabolic capabilities. So we're, in a way, extremely under-evolved when it comes to that. By contrast, microbes are able to generate their own organics. Microbes are able to breathe oxygen, but oftentimes many other alternative final electron acceptors. So that's a fancy way of saying that they can breathe things that are not oxygen. So when the microbes find themselves buried in sediment, there's billions of them per cubic centimeter often. So this billion microbes breathing the little bit of oxygen that's penetrating into the sediments basically exhausted completely, and then they have to look for alternative things to breathe. Usually when oxygen is depleted, they move on to breathing nitrate. When nitrate is depleted, they remarkably can move on to breathing manganese. They can breathe iron. They breathe sulfate. Sulfate's a pretty big one, so it's a pretty big source of a final electron acceptor for a lot of microbes that live in the mud, especially. We can tell when we've gone to a sulfate reduction zone in the mud because just smelling the core, if you were to pick up a sample of mud, it smells like hydrogen sulfide or rotten eggs. So that's microbial breath. They're breathing that out. Again, just like we're breathing oxygen and then breathe out water, in our breath. Microbes that breathe in sulfate breathe out hydrogen sulfide as their breath. So we can definitely smell that one. And when things get really bad and sulfate is gone too, they completely have exhausted that resource. You've got some pretty nifty microbes called hydrogenotrophic methanogens that are able to generate methane basically by having used carbon dioxide, a horrible oxidant, not a very good oxidant, but they force themselves to use that carbon dioxide and as a product generate methane. So a lot of diversity in terms of the I should say this microbes are extremely evolutionary advanced. It's hard for us to see that due to our macrobiological bias that we tend to associate evolution with the change in form of animals. And perhaps change in behavior, right? Like cognitive behavior, if we're talking about early Australopithecines or primates. But in the world of like hardcore evolution, coming up with something truly novel in terms of operons and genes and functions to interact with your environment or metabolisms, again split up into anabolism, meaning how food becomes part of your physical makeup, and catabolism, meaning how you manage where your electrons come from and where they go. In terms of all of that, we're safe saying that we're not very evolved as animals, and specifically humans, relative to the microbes. These are highly evolutionary advanced life forms that clearly people like, again, Julie Huber in the room and I have a lot of respect for. Yeah. These things are cool. So yeah, we're continuing this filtration step. This filtering step can take up to 2 hours. So it's a very exciting 2 hours. Again, for our Woods Hole oceanographic microbiologists here that have been working to engineer this machine for years, I suppose.
Someone else invented it.
Okay.
We just retooled it.
All right. Was this Greeks from Hawaii?
What?
Who worked on this first?
The scientist engineer who invented it, his name is Chip Breyer.
Chip Breyer.
He used to be at Hui, but now he's at University of Texas, Rio Grande Valley.
Okay.
It's the valve that's really unique.
So a bit of history behind the engineering of this filtering apparatus that we're using here. Yeah.
Is it possible to turn down the ISO a little bit and increase size? It's a little bit pretty.
One more time.
I'm just asking the pilots.
Oh, sorry. I thought you said Gus.
modify
Yeah.
the-
Okay.
No worries.
Yeah. So again, the idea behind doing these filtrations is to catalog the microbial composition and the activities of the microbes that that are emanating from the subsea floor crust at these sites of venting hydrothermal fluids. These fluids will carry with them a unique microbial community that's drastically different from the microbes that are found in deep bottom waters around us. So yeah, we try to get this probe exactly where the warm fluids are emanating to minimize or completely exclude, to the best of our ability, background water infiltration.
Gus, we have a question in the chat that's saying, do you have a favorite microbial metabolic pathway?
Wow. I'm gonna go for hydrogenotrophic methanogenesis. It's my favorite
Okay.
right now. Yeah.
Say that 5 times really fast.
Hydrogenotrophic methanogenesis. So yeah. So again, food is more or less food is more or less a source of electrons, right? Sometimes those electrons come with organic carbon for you to, on top of getting energy from the food, be able to incorporate that organic material into your physical body, whatever that might be. You know, that works for humans and for bacteria and archaea. Yeah, with this hydrogenotrophic methanogenesis, this specific metabolism is performed by a very special kind of microbe called archaea. We used to think that the archaea were rare, but turns out that they're not really rare. They were just extremely understudied. The way that we were looking at diversity in the early days of something called PCR and 16S gene sequencing was kind of excluding them. So making a couple of corrections on our technique for surveying diversity at the molecular level allowed us to see that there's pretty much archaea everywhere. They used to be very highly linked to the word extremophile, but as far as I'm concerned, everywhere that we've looked, you know, they're there. Now with these less biased tools. So anyway, yeah, these hydrogenotrophic methanogens are able to basically take electrons from hydrogen gas, or H2, and break that up, and then get energy as they're breaking that up, remove electrons from hydrogen, and ultimately reduce CO2 into methane. So I find that exciting for a number of different reasons. This activity leads to the production of a potentially extremely harmful greenhouse gas. So the methane's got to go somewhere, right? And sometimes the methane begins to want to leave the sediments, but there's a layer of other microbes there that can anaerobically reoxidize methane to make sure that it doesn't escape into the water column and eventually the atmosphere, which would be pretty bad. That would be a runaway greenhouse effect. So yeah, I feel like these communities of hydrogenotrophic methanogens and anaerobic methane oxidizers in the sediment are probably some of the most interesting things to think about. And again, the fact that there's microbes making this extremely potent greenhouse gas in the sediment, right, that can trigger a runaway greenhouse event is quite noteworthy, because these activities and the balance of these activities, right, including the microbes directly above them that are responsible for keeping the methane in the sediment and not letting it escape to the atmosphere ultimately, they played this balancing role that's probably modulated climate on our planet, at least modulate the temperature of the planet over geological timescales. So that's So yeah, like how that little, how that metabolism, specifically hydrogen-trophic methanogenesis, fits into this larger picture of geological timescale climate modulation on our planet is why I'm so interested in it. So that's a long-winded answer to that question.
No, Julie's not here anymore. I was gonna be like, Julie, what's your favorite?
Julie's not here.
I'll ask her when she comes back.
Pretty strong follow-up in terms of my favorite metabolism. My second favorite metabolism of microbes is probably something called microphilic iron oxidation.
Okay.
And it's basically microbes breathing out a mineral called an iron oxyhydroxide. So their breath more or less becomes this physical rock that they have to extrude, like right on the surface of their cell wall, so you can see them in the microscope. And again, they do this in places where there's very, very high quantities of iron being present in the water column. So one place that I can think of where these things are notorious is Loihi Seamount. So I've got a couple of friends that have been there, picked up rocks from there, and you can see the twisted stalks in electron microscopy slides of the rock. And the twisted stalks are literally a record of breath, solid rock breath that these microbes exhale as they were growing. And you can track the stalk too. They grow to a certain length, maybe 10 micrometers in length, and then you can see a splitting point, kind of like a split end hair. And that splitting point shows the physical time record in terms of solid breath of when that cell underwent a split and divided into two.
Wow.
So it's a remarkable thing to look at. And it's basically a fossil that we can find today of activity of microbes, right? Like very intimate activities is again what they're breathing out. That can be very old sometimes too. So sometimes that activity is over, there's no more iron there, but you find all these twisted helical stalks in the rocks, and you know that at some point it was loaded with iron and a little bit of oxygen. And again, those guys are called microphilic iron oxidizers. And yeah.
Philic iron oxidizers.
Yeah, and like the picture child for that is a pretty cool bacterium called Mary profundus ferrooxidans. So that's my second favorite metabolism. But I go back and forth. It's a weird question. It's like asking who's your favorite beetle, you know.
Yeah.
Yeah, there's no real answer. You know? I love them all, you know.
Yeah.
Yeah, I'm on a Ringo kick, obviously, right now. Yeah,
As you should be.
as I should be, because of the name of this cruise, of course. Yeah,
Yeah.
that's right.
I'm shocked that I've not heard
Yeah.
anyone play Octopus's Garden this entire cruise.
Yeah.
What are we
point.
doing?
I don't know.
Well, we can't do it live, but we can do it on our own time.
Yeah.
You and I we can jam that tonight. Yeah, I've been working on the intro.
Have you actually? Is that what you
Yeah.
were jamming to last night?
Last night?
Yeah.
I had a little session, you know. I think, did I sneak in Octopus?
No, it was
Start?
2 minutes ago.
What?
It was
No, I
seconds
was
ago.
playing a little bit last night.
Oh, really?
Yeah, I just snuck in like a 5-10 minute thing. Yeah.
Ah.
Yeah.
We also have a really good question in the chat. Are fossil fuels a renewable resource made by subseabed organisms rather than squash dinosaurs? So what are fossil fuels really?
Yeah. Okay. I think fossil fuels can have a number of different origins. So it's just it could be organic matter from like ancient forests that have become subducted to some degree, right, and like aged to
255.
a mush of organics. I guess biofuel, methane could definitely be biofuel. So this crazy metabolism that I was talking about, again, methane can be generated a couple of different ways, of course, but my favorite way that it's generated, I just discussed, called hydrothermal methanogenesis. And yeah, if we were to want to get methane, right, which is a pretty good gas for combustion, right, we could have microbes make methane for us, and I'm pretty sure this is being done somewhere already. But yeah, we can have a microbe-derived biofuel for sure. And are they renewable? Yeah, I suppose that these are renewable. I'm just not sure what the timescales for regenerating the energy source are. so-
Probably very large ones.
Right, and they all ultimately lead to CO2, right, going back into the atmosphere. So I'm not sure. They're perhaps renewable, but it's I'm not sure at all, and if they're going to be green sources of energy. That's for sure, because we end up generating, repopulating, or adding CO2 or carbon to the atmosphere at a rate that it would that carbon will find itself back into the atmosphere naturally. So that's a real issue here.
I think I want to circle back to what you said about microfossils at Loihi Seamount. So these microbes that you're studying that live in the crust, how big are they? Like, are they a couple microns, fractions of a micron? Like, what's the size scale that we're talking about?
Yeah, I think that as far as I'm concerned, everything's going to be at around 1 micrometer in length. Yeah, 1 to 2 micrometers, I would say max. So definitely, these are on average the smallest living organisms. I guess viruses get a lot smaller, but of course, like it's an open question as to whether those are living biological entities or not. You know, we're not going to open up that discussion right now. We would need a full 5-hour podcast format with both sides being represented equally before discussing whether or not viruses are alive on this.
Hey, we can make it happen
yeah.
at some point.
Yeah,
If
yeah.
you want.
Oh yeah, I want it. And let's just do it in a cage, because it's gonna get ugly.
Oh no.
Anyway, another remarkable achievement, actually performing this dive, has been sampling really old mud that for some reason at this very interesting geological formation sits right at the seafloor. So I guess this is something that I'm doing in collaboration with my colleague Maria Sandoval from the University of Costa Rica, and she might be able to better explain the geological background behind this. But we basically, as far as I'm concerned, there's ongoing discussions between petrologists and geologists in the ship as to what's the mechanism that's leading to this extremely old mud rising up right above the sediment in some places at these sites called escarpments. So yeah, long story short, mud is basically a microbial time capsule. If you have an uninterrupted sequence of mud, meaning that there hasn't been a catastrophic disaster on an adjacent seamount that leads to a rock slide that disturbs the mud sequence. If you have a perfectly preserved sequence of mud, you can generate a model based on how fast the never-ending snow that we see here, basically organic matter, how fast organic matter pellets to the seafloor. And these models have been generated from many different places across the ocean, so we have a pretty good idea of how far back in time we go as a function of how deep into the mud we are. So anyway, yeah, so mud is a way different system that's also considered the subseafloor, but it's extremely different than this very active aquifers that are made out of rock where water moves. And I would say on average, there are hundreds of meters of mud above the natural flow path of these aquifers in the bedrock beneath the mud. And again, the water in the aquifers in the bedrock beneath the mud is completely different. It's literally a different physical, chemical, and biological ocean than the ocean above it, on the above the sediment layer that's covering that bottom ocean. So yeah, there's like this two physical layers of ocean. Again, one in the crust then there's a ton of mud above the crust, and then there's the seafloor that we can see, and the seafloor that we're landing on that you'll see on your screen here. Anyway, so going back to the mud sequence, on average, I would say abyssal plain sediments and like the middle of ocean basins can be 1 to 200 meters in depth. And the sedimentation rate of this thing varies a lot. If the tectonic plate is under the gyre, in the middle of the Pacific, the South Pacific, for example, it's not going to accumulate a ton of mud that fast. So going down 10 centimeters into the mud may bring you back 100,000 years or perhaps 1,000,000 years, in like just a few centimeters of mud beneath the seafloor. If you're really close to the sediment, that time relationship to depth changes drastically because, and I meant to say really close to the continents, yeah. So when you're really close to the continents, there's so much organic matter coming in to water that's near land that there's fisheries, there's fish defecating, there's fish dying, there's organic matter being produced. So that layer builds up extremely fast, and it might take 10 meters to go back, 100,000 years or 1,000,000 years. So there's a weird relationship between where you're at in the world and how far back in time you've gone once you get into the subsea floor as a function of depth. So anyway, long story short, once those models are worked out, then you can be pretty confident that you know the age of the mud layer that you're working with. And anyway, long story short, it seems like some of the oldest mud on the Earth is actually found on the Western Pacific Ocean, so the other extreme of like where this entire ocean basin goes towards Japan, and it can be 10 kilometers thick in some places, and the bottom of that mud layer can be as old as 230 million years old. So yeah, long story short, the mud is a microbial trap. Nothing can move into the mud or out of the mud at a rate faster than molecular diffusion, which, again, this in physics, this is called Fick's law. So it's very slow movement. So microbes basically become incommunicado. They become, a lot of them die, obviously, the first 10,000 years of entombment, right? We can see cellular densities dropping by 4 orders of magnitude, meaning that once a particular mud layer is older than 10,000 years, the number of microbes that are there are about 0.1% of the number of microbes that were there at time 0. So most of what's happening in the first 10,000 years of entombment for this microbial communities is just massive die-offs. So this opens up a treasure trove of evolutionary ideas. Which microbes survive? Why do they survive? It's basically a mass extinction at every 10,000 year depth increment that you're playing with here with this muds. And again, if you go to the right places to dig really deep muds, you can study that experiment of what happened to these microbes that were completely isolated from the water column, the food sources of the water column, and the oxidants of the water column for 230 million years. So that's another ongoing active field of research in my laboratory. We're very much interested in microbial evolution in diffusion limited environments, meaning microbial time capsules that are represented by microbes getting trapped in the sediment and having nowhere to go. But I should say this, at least to like end this stream of thought, but they managed to remain alive.
Yeah.
So if you go all the way down to 100 million year old mud, there's a pretty good chance that at least a couple thousand cells per cubic centimeters will be alive, and will have not just adapted, but actually evolve, meaning change their distribution of alleles to provide them a selective advantage in the subsea floor. The mechanisms for doing that, how that's happening, at what rate that's happening, what lineages this is happening are still being studied. And again, this is an active area of research in my laboratory too. So deep time in evolution. It's an extremely fascinating prospect for that wants me, or that leads me to continue to want to study the deep subsea floor.
I have a bit of a selfish question because something that really interests me a lot is really cool organic compounds that microbes in the ocean synthesize. Are there any cool, maybe like potentially novel, like organic compounds that we're studying that could maybe have like, I don't know, medical applications or anything like that? Are there any cool, I guess, compounds of value that these microbes synthesize?
Yeah, I would say absolutely. And again, I'm most familiar with this question in the context of marine animal microbiomes.
Yeah.
So, yeah, I've done some work with Mediterranean sponges. And we've got a number of metagenomes that we've isolated from the osculum of a marine sponge called Aplycina aerophoba. Yeah, this was done with my my group in Haifa University, led by Dr. Lars Steinler. Shout out to everybody in Haifa. Yeah, so I really focus on something called the metatranscriptome. So again, the messenger, the real life messages that microbes were sending to each other for most of the work that I did there.
Close up on the rocks.
A close up on the rock, sorry. And I'll come right back to that story. Let's see.
All
Which
good.
rock? The one, the escarpment?
Yeah, I guess so.
Is that Maria?
Soda.
Oda, okay. If it's soda, she must want to look at this. Let's see. On the left side, okay.
So if we get into port morning of the 15th, is that right?
Sounds right.
I can check.
Just trying to decide if I can do another experiment. Because they run, I mean, I can trim it back, but they normally run a full 4 days, which I know I don't have. So I'm just trying to decide if it's.
Yes, we get back to shore around 8:30, 8:45 on the 15th.
Okay.
Yeah. But again, that could change. You never know. But we'll see.
Well, we're not going to be craning off gear until that morning, so that's what that's
Exactly.
like my benchmark for. Got to be done by then.
So our collaborator from the University of Costa Rica, Odalisca Bridi, Professor Odalisca Bridi, has requested a close-up of what I'm pretty sure is the animal life that lives on top of these rocks. So that's what I'm doing now. Oh.
Oh, wait, today's the 12th.
Yeah.
Today is a 12th.
Jesus. there. You could tell them how we're using to communicate on the ship, Gus.
What was that?
You could tell them, like, it's pretty unusual to have be able to use WhatsApp effectively. The internet on the ship is so good that we actually can.
Oh, that's right. For those of you guys listening online, yeah, we have incredible internet on this ship. I guess as would be expected with the last name Schmidt, I guess associated with it. Pretty high bar for everything internet related. So yeah, all of the scientists we're on a WhatsApp group, and we communicate with each other all the time. So no matter where we're at in the ship, there's usually a TV showing high resolution images of everything the ROV is up to on the seafloor. So yeah, one can even be in one's room, in the mess hall, in the laboratory, and if at any point on the screen there's something of interest to you scientifically, you can just, as a scientist, you would just send a message to the people that are working at the control station for the ROV, and we will be able to accommodate your requests. So that's more or less what I'm doing now. I am accommodating a request by Professor Odalisca Bridi from the University of Costa Rica, who wanted me to have to pan the camera around to look at these animals on the rock. She is a octocoral specialist, and yeah, so some of these views are making her quite happy. I guess I'll do a little more over here. No animals here, so this is probably not what we want to look at. Perhaps just that. Right there.
We haven't seen any octopus on this dive, have we?
Not that I'm aware of, no.
Okay, well, I was happy with my survey.
Yay.
Fun camera work.
Yeah.
Stuff us.
I guess at this point in the yeah, oh wow, 6 minutes that I have to continue narrating. Yeah.
Time flies when you're having fun.
Exactly. Yeah, time flies. Yeah, we need the 3 to 4 hour formats again. Yeah, we'll do that podcast style next time. Yeah. So yeah, going back to the useful compounds, potentially valuable compounds for society too, made by just microbes. Yeah, again, that question often comes up, and again, I know I've heard that question too, but again, mostly in the context of microbes that are making special compounds in association with animals. So yeah, some of the work that I've done in the Mediterranean involved sampling metagenomes and metatranscriptomes from a shallow water marine sponge. So this sponge hangs out just 5 meters underwater usually. So it's completely bathed in the sunlight half the day, called Aplycina aerophoba. And yeah, from looking at that and using molecular techniques to predict something called secondary metabolites, which are, again, organic compounds that are not necessary for the microbiome of the sponge to stay alive, but necessary to perform a function with the environment that confers an evolutionary advantage. That's that's a technical definition of a so-called organic compound. Oh, sorry, a secondary metabolite. That are all organic compounds, of course. So anyway, yeah, these things are predicted to be treasure troves of really complex, again, secondary metabolites.
I was going to return it because that didn't take
Yeah, secondary metabolites have been shown or have been experimented with for as therapeutics for different medical ailments. Marketing products too. So I know that at least in the project that I was involved in,
sitting in anything?
this consortium of microbes basically
Definitely like stirred up.
cycled carbon, specifically methane-derived compounds. They cycled that back and forth with different species to ultimately generate a secondary metabolite called
Yeah, so.
lycopene, that basically has UV absorption properties. So at the end of the day, what these microbes were up to, you know, this or one of the functions of this very complex microbiome that was living as
10 minutes.
a thin slime layer on top of my sponges, they were all working to make that animal sunblock.
That's insane.
So that was a brand new, I guess, a multi-lineage symbiosis loop that was never described before. That I guess I'm putting out a selfish plug there that we put out on our paper published in the journal Microbiome about 8 months ago.
Cool.
So yeah, microbes will make sunblock for their host, and of course that's in terms of applied microbiology, we can always capitalize on those microbes and have them make that sunblock in an industrial setting for us too.
Yeah. What would be required, I guess, besides like investments?
Oh, I mean, we would need a very defined culturing strategy. If we wanted to like exactly make microbial sunblock for marine sponges, right, in the laboratory. So we would need to know which one or which lineages are involved in each one of the transformation steps for these microbes. And we would have to provide them with the appropriate conditions, and we would have to do this in very large batches, so managing the redox condition of the batch. This is basically industrial level microbiology. It can be done, right?
Yeah.
It can be done. We make yogurt and beer,
Oh yeah.
and we've done it for a long time, even way before the golden age of microbiology with Louis Pasteur or Alexander van Leeuwenhoek looking at the first microbes thousands of years before that. I believe that if you go to the Cairo Museum, the oldest papyrus that exists, like from some of the first dynasties, like 5,000 years ago or more, are just instructions on how to brew beer from barley. So microbiology, against my biased point, microbiology specifically how to manage this metabolisms, right? Fermentation, managing microbial metabolisms is what's led to civilization, you know, in my
Yeah.
the need to have year-round access to beer, to barley beer. It's basically what led to the Egyptian civilization. You know?
Pretty much.
So yeah, it's all microbiology to me. I see history through the lens of microbiology.
I mean, it's important. Also, we wouldn't be alive without all the microbes that are helping us. So.
Right, and we wouldn't have cheese, and we wouldn't have wine, and we wouldn't have beer, which is
Or bread.
which is
or
the reason why
chocolate.
a lot of us live actually. All right. On that note, I'm gonna hand the baton over to Dr. Jorge Cortez from the University of Costa Rica, the co-chief scientist of this expedition. So, all right, welcome, Don Jorge.
Thanks.
Thanks, Gus.
Buenas tardes, buenos días, buenas noches. Les habla Jorge Cortés de la Universidad de Costa Rica y voy a estar aquí acompañándolos durante la próxima hora en esta expedición a 200 kilómetros de la costa de Guanacaste. Y en estos momentos estamos realizando un muestreo. Tenemos una sonda dentro de una caverna que encontramos en este sitio, Monte Submarino Tengosed, y se están tomando muestras ahí y vamos a estar aquí un rato tomando datos de temperatura y oxígeno. Y mientras tanto vamos a ver los animales y vamos a hacer algunos acercamientos ahorita. Good morning, good afternoon, good evening. This is Jorge Cortez from University of Costa Rica. Right now we're sitting here for a while. We have lower sensors into this hole in this seamount Tengosed Seamount, and it's collecting water samples, and we'll be lowering later also temperature sensors, oxygen sensors, try to understand what's going on in there, what's going on deep in these rocks.
Well, just seawater in this case.
And,
It all goes down with milk.
And in a little while we'll
The chief originally designed
try to make some close-ups of these interesting animals we're looking. Sea fans, anemones, sponges. And here with us
5 meters above the black sand, 10.
in the room is some of our other colleagues from different institutions doing different works, doing research projects here.
So it has a brain.
Entonces vamos a estar aquí unos minutos más y nos acompañan en la sala personas de otras instituciones. El que estuvo conversando, el doctor Gustavo Ramírez, colega de California, especialista en microbiología, y ahí lo pude oír dando explicaciones muy completas sobre la microbiología.
Or get trained. It is possible, like I could do it for more people. Look around.
What's the fixation mechanism? Is it just filter salt or?
And I just played it with
Filters
some
have a reservoir of
close-ups.
emulator.
Yeah.
Of course, and then there's two little holes.
That's what I like.
I know.
I know.
I've always done it. It seems to work.
Yeah, yeah.
I know some people don't like it, but it works for us.
Okay, so
I
let's
do
see
have
how
some trouble with solid matrix.
And it's always worked for me.
Yeah, I don't know. Some of the sediment people
Anyone
don't seem to like
that's
it.
got something to say about why we're using it here, I don't think understands what the conditions under which this is happening.
Yeah,
It's miraculous we're here to begin with.
right.
Yeah,
It works for me.
Get nitpicky about
Yep, exactly.
but RNA later, which is another product.
Yep.
Esto que vemos es una anémona de mar, detrás un coral negro, a la izquierda lo blanco es un octocoral con una figura, y lo que parecen ahí unos hilos, eso es un coral negro, me parece. Lo que parecen unas rayas blancas sobre la roca, esos son poliquetos, esos son gusanos.
Find RNA
Y vemos ahí algunos otros.
pretty easy to work with.
If there's a ribosome, there's RNA, right?
Yeah.
So there
Vamos
should
a
be
poner los láser para ver el
Yeah,
tamaño.
getting the message can be hard.
Here's the anemone,
North Holland is probably the hardest place.
and at first center, behind that the black corals, octocorals around, and the white wiggles that we see on the rocks, those are the tubes of worms.
So we like did first strain synthesis, second strain synthesis, and then made an idea from that.
Nice.
And it worked.
This is forest pretty.
Okay,
So that's
there's the lasers, those are 10
In
centimeters
the North
apart.
Atlantic, it's very similar.
So that anemone is something like 20 centimeters. Y los láseres están a 10 centímetros de un punto del otro, así que esa anémona puede tener unos 20 centímetros. Estos láseres los usamos precisamente para poder tener una idea del tamaño de lo que estamos viendo.
Almost there, Jeff. Almost there. 3 minutes left. 2 minutes. Sorry, 2 minutes left. Thanks, Gus.
Hoy siguen esa octocoral que estamos viendo, es un octocoral grande, rodeado de corales negros en su mayoría. randies. They're very, very small. Very hard to collect except if they're attached to a rock. We can pull the entire rock out. We have
Alright, stopping that sample.
nice composition of black coral, octocoral, and ophiuroids.
Alright, we can take that intake out carefully.
Randy wants to know if we can fire an Iskin bottle while we're sitting here.
Right now?
Yeah.
Okay.
So, anytime, any one of them should do it.
Okay.
So it's Randy's request.
Yeah, we'll
Okay.
do that.
Sorry, she texted me and I completely forgot.
Well, we'll pull this out
Okay.
and do that.
Yeah, we'll do it now. Just so we don't kick up the desk when we're pulling this
The
What are we doing?
nesting.
Randy's
Excellent.
requesting Iskin. Does that work or is this?
Yeah, I think we, oh, yeah, no, it's fine.
It works? Okay. Thank you.
Yeah, we'll take that water sample and continue with this.
Okay.
Let us know which bottle. Right now we're recovering experiments and this will take a while and we'll pull down other sensors and then we'll continue looking at the seascape and organisms here. Ahora vamos a remover los sensores que tenemos en esa caverna y vamos a poner otros y después vamos a continuar viendo los paisajes.
That was supposed to be for resin testing, but I don't think we need that sample for that.
Alright. Now the fun begins.
I don't know, it was pretty fun dropping it down the hole.
It's always fun dropping down the whole thing and back. Yeah. You happy there? Yeah. I'll just fly out
Here we go. You can see the hose that was lowered and trying to get it out there now. Sí, con esta manguera que están viendo ahí se ha estado bombeando agua. Sí, es un muestreo muy especial. Ahí viene el sensor. It's looking good. Ahí está, esa es la bomba que ha estado extrayendo agua.
was an Octopus Odyssey. What's this?
You could see the hose where the pump was sending the water, and we have these incredible samples from way into those rocks. Hmm.
Just pull it up so you can see the main camera. Just leave it back. As far as
How long is the hose?
25 feet?
5 feet. It's about 8 meters. Sí, la manguera que vemos aquí tiene 25 pies, que es más o menos 8 metros. Entonces sí es una muestra, pueden ver lo profundo dentro de la caverna.
do you want to try to put it in there, or do you want to just lay it there? I'm just gonna put it right next to it.
This hose is 25 feet long, that's about 8 meters, and so you can see how deep that cavern is, and we have these samples are going to be very interesting because this is very pristine water from way down into the rocks. We're seeing some sea pens right next to the instrument. Aquí lo que vemos son esos plumas de mar, varias plumas de mar a la par del instrumento.
Hey Jorge,
Yep.
for those 2 seamounts, why not something, and forgive me if this is culturally not appropriate, but like amigos? Wait, doesn't that mean friends?
Yeah.
Because someone was saying that other word, mantu, or whatever, is singular friend,
Yeah. What about
amigos is
those like
plural.
a basket? Maybe it's that. Just like
Okay.
the basket on top, and you put the sensor on top of the wings, and they kind of play the name.
Yeah.
That's like
You want to fall over? You can grab that.
I have one team for a lot of.
Yeah, I think I make
Yeah.
yeah, I like that.
Well, the history behind it, from what I learned, like in terms of the language, so.
Okay, that's from the Mayas.
Yeah.
That they reached the furthest south they reached was around 830 Costa Rica. Right now, that's your.
Yeah. Is it just a hose on there, Jeff? What's that? Is it just a rubber hose? Yeah.
Hey,
there's a
just
hose
checking
with,
in.
Yellow line. Are we okay just to grab it? Oh, yeah, you
Our
do
scientists are
crush it.
currently doing a little brief,
So you're just
and I
heating
just
that
wanted
up.
to check in and say hi, ask if anybody had any questions for our scientists.
This would be.
Are we going? There's a question saying, are we going back to Fuentes?
This one's, how do you say?
Not that I'm aware of. I only have the dive plan up to the next dive, which is dive 630, and dive 630 will be on Dorado Outcrop, and that is launching later tonight. So you get 2 locations in one day. So that's the plan for today. Yeah. So let us know if you have any more questions. I'm happy to answer any as well while the scientists are busy doing their science. So yeah, type them in.
Oh, no, by all means. By all means. Yeah, I was talking earlier.
We have a question saying, is that the last dive? I'm not sure. I heard rumors that we could be diving on Wednesday or Thursday as our last dive, but that has not been confirmed yet. The scientists need to figure out what their priorities are with the time that we have left.
Yeah, so right now the milk crate is has a couple osmo samplers in there. Means they've got an osmotic pump and they've got a small bore Teflon tubing, and so the hose was designed to fit down into the hole, and now we're trying to put things back. So inside there, you can see inside the milk crate, you can see a pump with a little red top that has a membrane in there that separates salt solution from a non-fresh solution, and that drives the osmotic pressure gradient across the membrane, which causes it to pull water into the small bore Teflon tubing, which we will, once it's recovered, we'll cut that up and put it in little vials and analyze it back on shore. The other thing they're picking up now is an intake to the water sampler, and that also was long to make it down into the hole. And once all this gets cleaned up, there'll be a third instrument, which is a dissolved oxygen instrument that will be lowered down into the hole, so we can get an idea of what the concentration of dissolved oxygen is of the fluids that are down there. So as they're putting all this away, they're putting the tubing back.
Ese que habló ahora es el doctor Geoff Witt de la Universidad de Alaska, y está en California, en Moss Landing, y lo que explicaba era de lo que está en la caja negra allá. Eso lo instalamos hace una semana y ha estado tomando muestras de agua. Es un sistema muy ingenioso para ir tomando pequeñas muestras de agua por medio de una presión osmótica. Entonces saliendo agua dulce y va entrando agua de mar de esa caverna. Posteriormente se hacen toda una serie de análisis químicos de esa agua, y eso nos va a ayudar a entender el origen de esos líquidos que están circulando ahí. Y en este momento estamos recogiendo la manguera vimos antes del otro muestreador, y vamos a después poner la caja y posteriormente vamos a bajar un sensor de temperatura y de oxígeno por unos 10 minutos. And here with us is Adam as pilot, and Andy as co-pilot, and Sophie with the outreach.
Hello.
Nos acompañan ahora en este momento Adam, que es uno de los pilotos más experimentados, y el copiloto, el que está manipulando el brazo ahora, es Andy. Y en la parte de comunicación está Sophie. Son operaciones de mucha paciencia, porque eso lentamente hay que ir haciendo todo el proceso con mucho cuidado. Aquí hoy no estamos tan profundo como otros lados, aquí estamos a 2000 metros de profundidad versus 3000 en otros días. So now we're putting the hose in there. We're going to put the box on top to hold it, and yeah, it always takes some time to manipulate everything.
you shake it?
Sure.
I get any sediment? There, it doesn't look like there's much on there.
We landed it in the dirt too. We know to pick it up, so it could be from that.
Okay. Thank you.
Looks
Nice and
clean. Looks clean. Looks good. Okay. Wow. Oh, that's good.
Nicely done.
Yeah.
Beautiful. Okay, we have all the instruments on board. Now we go for the temperature and oxygen sensors.
I'm starting with number 7.
That would be sample 257.
Just push down on that.
And this is just background, right?
Yep. Jeff, do you want background water for chemistry?
Yeah.
Would be a whiskey bottle.
No,
Oh,
she's
I'm
lucky.
filling one right now.
Okay.
Yeah. Trying to keep it consistent in the method, how it's getting collected.
Okay.
Plus, unlike Celeste, I don't think I can stand up there and like collect all that water.
Okay.
She's more patient than me, maybe.
Yeah, we still need to take the whiskey and we're being taken.
Oh, for Randy?
Yeah.
I think you have enough and you have it in your hand.
You took one at the beginning of that, right?
Yeah, there's a Niskin bottle 243 and bottle 1.
So Jeff. Yep. So you want to just drop this in the hole and?
Yeah, drop it in the hole, wait 10 minutes, and then pull it out.
Okay. Is it how many feet of rope do you have there? Do you know?
I think there's 12 feet.
12 feet. Okay. Did you want us to like fly it? You can get over and drop it, and then we can kind of maneuver it. Yeah. Yeah. Let's get up high and drop it. Do you want to leave it on the bottom and fly away from it? It should keep it up, or you want to just drop it from high?
Should we put on the overlay, Jeff?
overlay for this, it's already independent from.
Oh,
And if you grab the end and you start to feed it off.
It's not the Andara probe, it's the
oh.
overlay.
So we won't get the data back till they download it.
Yep.
Ok. Vamos alistándonos para bajar este instrumento en la caverna, y el instrumento este va a medir oxígeno y temperatura. Ujú.
I guess maybe I'll just fly up. You want to hold it? Probably even fly up. Actually, yeah, just grab a hold of somewhere so you have some visibility over it. I'm gonna grab it.
Muy bien, ya tenemos el instrumento y vamos a instalarlo ahora. Aquí ya vamos viendo la caverna. There's the cavern where we're going to lower this other instrument. And as I mentioned before, we're going to have some really interesting data from down there.
That one doesn't have a name, right?
It's marker X.
Yeah. But like, no, just marker X.
Well, we have to change now. Coco is the co-pilot and John is there with the outreach.
Are you leaving anything?
And the
for
pilots is still
the
Adam.
Julia?
I don't know. Because we went through what she needed before I left,
Mhm.
like made sure that we had it in the lab. And I haven't, so I don't know. Nothing specific. Originally, it was gonna be my pup. That was the only thing I was planning on.
about 257.
Just because we can.
Do you want it to be further down?
Is that-
Look up at my set. So if you do that, now if you can just angle some stuff in this direction. Just want to get that handle wedged in that crack, so even if you come up with that big
Mm.
place. Yeah, and then just lower it. Isn't it to drop it in there, because the sensor ends
Right, yeah, if you lower it in and maybe lower it to the, on the right side of the crack.
That's pretty good.
Mhm. Ahí va bajando el instrumento.
Are you happy at that depth? Or you want to go all the way? The only reason I say this is that handle, I worry about getting caught in there. Because it's something we can't see.
Okay, yeah, no, if you're worried about that, then let's just stop here and we'll hold it for 10 minutes.
That's not skipping the rope.
Yeah, it looks like it's over the ledge.
Yeah,
Yeah,
looks like
it's
it's, I mean,
over
it's
ledge.
down there, so.
Yeah.
Yeah, just freeze it.
So 12:48, yeah, 48,
Yeah.
58, 49.
Can you power off the super, please?
Did you want to take the camera home?
Sorry.
You want to take the camera?
Yeah. Okay, great.
Thank you.
Bueno, ya bajamos ese instrumento, va a estar ahí 10 minutos.
Under this ledge here.
Yeah.
H for that to go down and then.
Yeah.
Vamos a
That's a good
ver
call.
los organismos.
It's a good place for the tube, 'cause that's where the water was coming out.
I think it's sitting, so you're not actually touching the sensor. So
Yeah.
If there's
Yeah.
something below there,
Since I was a lead.
it hits that sensor. And in hindsight, we should have like put a pipe over it or something.
Yeah,
Now we're here now.
yeah.
Thanks, guys.
Yeah.
I know it was thrilling. Although you did get to put something down a hole and bring it back.
I think so. Yeah. You're happy with that position?
Yeah, that's great. Yeah, we'll let it sit here for 10 minutes.
What are we going to do ballistic?
Once we're done with this, we'll take the next one.
And Jeff, don't forget about the marker.
Margaret.
Marker. Alright, and you're going to bed?
I'm gonna move around
So
with
your Osmo
looking at the
bin, we should just put in the cold room?
and
Yeah,
we're
you
gonna
just
do
put it over by my desk.
Okay.
one like
And
this
when will we
and
see you again?
one with the laser.
Dinner time.
See one. Okay. Here we go. Yeah. One with What we're looking now is at a crinoid. Estamos viendo es un crinoideo como de 10 centímetros. Pueden verlo al lado de la anémona. O probablemente está más pequeño, como unos 5 centímetros. Lasers.
Bless you.
Bless you. Now what we have here in focus is a sea pen. Estamos viendo es una pluma de mar. Y otras cosas que vemos ahí son tubos de poliquetos, tal vez otra pluma de mar o unos hidroides. What we've seen here is a sea pen. These ones are interesting because they have this foot and seems like they can move around. And also seen polychaete tubes, maybe other small sea pen, barnacle. And this is probably a xenophile form. This is a foraminiferum. not very apparent, but when you do a close up,
Yeah.
you could see.
There's some slightly on the edge there.
Yeah.
Oh, yeah. Yeah. Well, it almost looks like it's slowing down.
Uh-huh.
One more minute.
The one with the lasers.
All right, yeah, we're all set here. Thank you.
All right.
Thank you.
Yeah, so you did review it.
And I got that just in time.
Just the other one.
So we still don't know what's really how deep is it, or not really.
What's that? No, it's fine. We'll just lay it in the nice soft sand. Look it down nicely if you can grab it. Just curl it up.
Yeah, I can see that.
Okay, so we finished with this sensor, and we're gonna pack it now, collect some water, and move on. Yep, vamos a terminar de guardar el sensor. Y apenas terminemos eso, tomamos una muestra de agua y empezamos el ascenso.
Just slide it on there.
You know he's tossing the milk crate.
It's floating line.
There's little weights on the floating line, or the yellow line, right?
Yeah. We have to put it in rate. Just pull over towards yourself. Figure it out. Even like that. No, you gotta do it again. Just finishing up. I'll see that. Everything going okay? Yeah. I would come down on it like this. Grab it. So you grab the T-handle and we'll put it in brake position and roll it up with the T-handle. So we basically got the other sample, the awesome sample, and the thing between on the hole. It's all stuck on the porch really nicely with the weight on top of it, so it's pretty good. If you're recovering, you may want to just like, especially once we figure this out, we'll probably feel bad about how long it's been in the military too. But just so none of that floating around for the professor or something, they might just stick it in there. Other than that, put the date on here.
We need a listing bottle before we leave this place.
And I think they may want to do timing. Let's check.
No, because it's we should be done by 12:15.
Right. Okay.
So we do the listing and then another one close to the surface.
Right.
So I think that
And
by the time you finish it.
and also we gotta deploy marker X, right?
Yeah, we have to drop.
Oh yeah, there's a marker.
What was that?
Marker X.
It's
Marker X,
it's not
deploy
great.
marker
Yeah,
X.
yeah, and then I was
Oh, yeah.
thinking, let's put it like, put it on the edge of the gradient, so that.
Where is that?
So if you bump up the
Crater by wood 0.3.
yeah, if you just go, actually, you just pan over this way, and just fly straight ahead, the crater's right.
Oh, it's straight ahead.
Yeah, it's like
Okay.
over here,
Okay.
like right over here.
Okay.
Yep.
Okay, so that's
There you go.
marker X. The other mission.
Good work.
Welcome. Good to see you here.
Thank you.
So we're almost finishing this dive. Now we have Doug as co-pilot. Coco is
Yeah, we'll.
handling the instruments.
We
And
just grab
at the
handle
end,
and put it on rate and see if we can't coil up the rope along.
oh, that's it. Trying to coil the rope and the instrument before putting it in the box. Estamos recogiendo este instrumento antes de tomar unas muestras y dejar un marcador y empezamos el ascenso. Y estaremos de vuelta en la noche.
That doesn't work. You can find it. Just take a whole rope with the weights and stick it in the grate and stick the instrument on top of it.
Yeah.
Mhm.
Yeah, almost vertical. Yeah. Oh, that's, yeah, that'll go to rate. Good job. Good job.
I was working like that now.
Yeah, well, there's something.
We're just stowing the probe.
Yeah, the really long.
Yeah.
I saw you guys holding it like this. It was like that.
Yeah. So stowing the probe, we still need to collect a Nesquin bottle at the bottom of the thing to us. And we need to deploy a
Now
marker.
you're gonna have to slow down and bring it closer to the porch.
And
You lose your slack, okay?
enjoy the Ascent.
You still got quite a bit though.
You do still have quite a bit.
Sounds good.
Even
And
after
actually,
free morning.
you porch out to meet him.
Yep. Face it down.
Yep. So that's last month. Or is it one speed?
No, it's free. It's not one speed. The more you turn, the faster it goes. Unfortunately.
I think even if he puts it around the jaw and he goes to drop it,
It'll fall through. It'll
Yeah, keep
fall
going.
through.
You're good. You're good. Keep going. Go.
Yep, you're good.
Good.
Look at that.
Good.
Alright, watch your tight.
Slow.
Start bringing it towards the porch. The porch is all the way out, bud. Yep. There you go. You gotta watch if you get it caught on something. Yeah.
Uh-huh.
There you go.
Remind
Start
me,
bringing it to the left. There you go.
remind me of making a sugar cone.
Yep, you should be able to dump it in the box now. It's plenty.
Taking out of this straight thing, so I don't
Yeah,
accidentally start.
yeah.
Hang on. There you go. That's just a pop up. Give it this ring thing.
Either one, Coco, wherever you got the most room. Looks like maybe in that right hand one. I'm trying to get this camera. There you go. Yep, right in there. Good, buddy. Turn your wrist clockwise just a touch. There you go. Perfect. There you go. You should be able to drop it right there.
Take that.
You can go on lock.
Yeah, it's on top. Start getting. There you go.
There you go. Beauty. Very nicely done, Coco.
Yeah, nice job.
Yeah, very good.
Yeah.
Bring in the porch. Or do we want to grab the marker while we're out here?
Yes.
Yeah, I can go ahead and stow that one. Or can you reach the marker like that? Yeah. You can hand over to Tyler. Okay. Thanks a bunch, buddy. Good job.
Get you a cinnamon roll, Coco.
Oh, there's cinnamon rolls. Alrighty.
Thank you.
Marker here. I don't know if you're gonna be able to grab it with that one. If you want to just grab it with your forearm. If you are, just still that one.
Nice job, Coco.
I got one.
Thank you, Coco.
You can try. I'll rack the camera back. Camera's all the way back. Looks like you might be able to grab it right by the decks there. Or by the top.
Mhm.
The old floaty bit? Yeah, the old floaty bit. If you want, you can kind of set it back down, let it float, and then grab it in the middle so you can have a good grab on it. Just, just be wary of, if you can pick it up like that, go for it. There you go. Okay.
So we're gonna set it by the crater.
Okay. You can kind of hold it out in the front like that, and I'll find the crater. We'll just set right down there. I'm gonna go ahead and torch in. That's fine. That's fine.
Now we're gonna leave this marker, and hopefully we'll be back here sometime in the future, and so that we can go exactly to the same places.
You might just have to direct me towards the
Que
crater.
ya viene a sustituirme.
Sí, si usted quiere.
Sí, sí, para es que necesito
Para que vaya a almorzar.
Sí, entonces ahora sigue el Niskin.
Ok.
Van a dejar este marcador en la cueva,
Ajá.
un Niskin aquí abajo y otro cerca de la superficie, porque ya se pone esto, se toma el Niskin.
Y para arriba. Ok.
Sí.
Ya no hay nada en la muestreo, nada. Ok.
Pero estamos sobre el tiempo.
Listo.
It's to the left.
Going that way.
There's like a
In front of me, behind me. Is anything else?
yeah, there's like a triangle in the rock.
Hand me back. Just this way, just a touch.
I did, because I couldn't go.
Try looking down with this camera. That will help out. Like here.
There's the triangle. There's the
Right here.
That's the
Okay, okay, I recognize that.
Yeah.
Where would you like this set? Like in front of it, behind it? Just tell me. Like right here to the right of it? Just like right in here on this little soft sandy patch? Okay, Tyler, if you want to go ahead and set it down here. Down, okay. You're happy right here? Okay. Yep, that's good. Spot coming down. Ready.
See you in 10 years. Let's do there.
It's fine.
Okay.
Beautiful.
And so now it's a Niskin.
Yeah, and try to get a brittle star. But we have time.
We have time.
Yeah. Yeah. Diva commented that we saw so many on the way here that looked different from the ones we got in all the corals.
Okay.
So
Seeing the science cameras.
Leo was like, yeah, it's different. So-
Okay.
Okay.
All right.
Are you on now, Sergio?
Yes.
Okay, perfect.
I mean, but if you wanna.
No, that's fine. Yeah, so
You're welcome.
a water sample here,
Mm-hmm.
and then just hop up and see where there were just these brittle stars everywhere.
Okay.
So we just want one, and we still have the coral equipment to put it in.
Okay.
This is where you'd like the, while we're sitting right here, you want the water sample?
Yeah. Yes,
Okay,
yes,
which
please.
Niskin would you like? There's like 3 maybe open?
Maybe number 3. Yeah.
Okay.
I think three's open.
Camera's wrapped back.
I'm gonna tell, because I haven't seen the specific ones, I'm just gonna tell maybe Diva or Leo.
I can pull up a picture.
Oh, really? Okay,
Yeah.
great. Thanks.
I'll go grab my computer.
Thank you very much to Don Jorge Cortez, one of the main deep sea scientists in Costa Rica. And I will join you for just a brief amount of time. My name is Sergio Cambronero. I'm a researcher at the National University in Costa Rica. And we are towards the end of this dive. We just have left a couple of more things. So what you're seeing right now is the sampling of a Niskin bottle, which takes a water sample at this specific depth. Yeah.
Yeah, so we're gonna see 1, 4, 2, 3. We'll go for 4.
4. Okay.
So just a real quick question.
Yes.
The other 2 are smaller.
Yeah.
So that will be one of the ones that's going to be taken at the surface. Then is going to be one of the smaller ones. Is that okay?
The one.
5 and 6 are smaller.
Okay. Yeah.
bottles.
Maybe we can leave them those 3 in any case. Maybe shoot those 2 at the surface and
Right. We
do 4.
Okay. Okay. So we'll go with number 4.
With 4.
Yeah.
Gotcha. Thanks, guys. Muchas gracias a el doctor Don Jorge Cortés que me entregó la coordinación de este buceo. Estamos finalizando nuestro buceo en este sitio
Or does it look like it wants to fire?
que se llama Tengosed. Es un monte submarino. Estamos en este momento buceando aproximadamente.
Yeah. 4 didn't fire.
4 didn't fire?
No. So
Oh.
we'll have to do it. We'll have to do 5.
Okay, let's do 5, yeah.
Try one more time. All right, we'll try one more time. It looks like that mountain's broken too now. Yeah, just be careful.
Les comentaba que estamos buceando aproximadamente 2000 metros de profundidad en el monte submarino Tengosed. Y estamos tomando una muestra de agua con un instrumento que se llama botella Niskin, que son unas botellas que tienen la capacidad de cerrarse y automáticamente capturar pues una pequeña parcela de agua, una pequeña parcela de agua a la profundidad en donde se encuentra la rubí. Entonces con este gatillo que pueden ver se activa y se cierran las 2 tapas de esta botella.
5 didn't fire either.
Didn't fire either?
No.
Oh.
All right.
All right.
Looks like the top went with the bottom. Didn't you see it right here? Where it's
Mhm.
still open on the bottom. There?
Yeah.
We can try 6, but then there won't be any for the surface.
Yeah, I think let's wait a
Try
little
5
bit.
again, but I don't hold out any hope that it's going to fire if it hasn't already.
Yeah.
Unfortunately, fired already. It just got stuck. What's that? It fired already, but it got. Yeah. Yeah. It did. It's probably the top is closed, not the bottom.
Yeah.
It's got stuck. Sorry. about
Stuck. Okay.
Yeah.
Let's just give me just a second,
Sure.
just to
We'll
double
get set
check.
up for 6 in case you want it. If not, it won't fire. Trigger it.
Beth, we had a small inconvenience. So we only the remaining Niskin bottles didn't fire, and we just have only one left. We have been able to take the water sample here,
Okay.
and Celeste is saying that she wants the.
Yeah, we saved the one for Celeste.
Really?
Yeah.
Okay.
Yeah.
Okay. Cool.
The one
All
for
right.
Randy was just an extra.
Okay.
Sorry about
Cool.
that.
It's okay.
Yeah. No props.
So this isn't the greatest photo.
Okay.
But you see they were just everywhere
Mhm.
as we were coming along. So
Okay.
once we come up, we'll just kind of look around to see
Okay.
if we see them.
Around those bamboo.
Yeah.
Mhm.
I don't even know. I mean, they were just everywhere. They weren't necessarily in association with them.
Okay.
But as we were
All
flying
around.
around, so, we don't need to spend a lot of time looking, but
Okay.
we'll just have a look.
Okay. Cool. That will go. Let me. So 3, 4, and 5 didn't fire. Okay.
Yes.
Just to make sure we don't. 3 was already fired.
3 was already fired.
Correct.
Already.
4 and 5 did not fire.
Did you know?
Okay, so we need to look and see where the Niskin was and why it didn't get lost.
Yeah. Okay, and I think we have one remaining. But
Correct. The 6.
do we have something on the bio box too?
No, that's still open.
Okay, so maybe we can put the real star there.
Yeah, or Coral Quiver 6.
I mean,
Or 8.
1 and 2 are recorded.
I mean, I would suggest if this is free, maybe to avoid the, that
Yeah.
it breaks.
Whatever you think.
Yeah. Okay.
We can use it.
All right. Perfect.
It just means we can't use it for anything in the water column.
Huh. Interesting. All right, now let's use coral quiver. Well, depending on the size.
Yeah. Although,
Oh,
yeah,
you-
I think you're right, because there was a suggestion to try to scoop them, right? Instead of pick them up
Yeah.
with the manipulators.
Mhm.
So maybe we have to use the bio box.
Yeah. Okay. Yeah, I agree.
All right. What are we up to?
So yeah, let's just go up a little bit and look around. We are trying to see, like, on the sediment, that is nearby, if we can get a brittle star, those that are like half buried in the sediment.
Yeah, so like back up on the ledge.
When you say up on the ledge, is it forward?
It's fine. Yeah, you're going the right way.
Okay.
So we came in from the port side.
Okay.
So if you want to just kind of look back over
Sure.
that way.
So we just finished with our sampling here, and we have one more remaining biological sampling, specifically a brittle star, which is also known as ophiuroid, and pretty much, yeah, we are interested because it looks kind of different of the ones we got earlier, and they're pretty abundant. So we're gonna keep our eyes wide open. Yeah.
Oh, it says they got the one. What is the one? Corals.
Can I pan?
Sure.
Yeah. Okay. Yeah. Nothing around here. Maybe if we go like over there, straight. Les comentaba que acabamos de terminar los muestreos en este sitio. Solamente nos queda una última muestra biológica de un ofiuro o ofiuroideo de que se conocen también como estrellas quebradizas. Y básicamente pues eso es lo que estamos buscando ahorita, y en cuanto tomemos esa muestra comenzaremos nuestro ascenso. Así que también pues les invito a que se mantengan atentos a sus pantallas durante el ascenso, porque realmente es el momento donde se ven también una gran diversidad de animales muy curiosos, que específicamente son los animales que se han adaptado para vivir en la columna de agua, y específicamente en los últimos o los primeros 1000 metros, de 0 a 1000 metros de profundidad, podemos encontrar una alta diversidad, como hemos visto en otros buceos.
No, that's not. They'll be pretty leggy. About that size, but like a small disc and long legs.
What about this ones over there? No.
I haven't seen one yet. Oh, there's one.
There's-
At the bottom of
yep.
the-
That
What
one?
are these two?
So in your screen, you see a couple of these brittle stars that we are aiming to sample. And around you have some corals. To the right, you have a xenophyophore. And yes.
Where are you at?
Oh.
Rocks too. Yeah. This is missing two. Sorry, I think we're missing. I mean, it has to be after.
Yeah. Do you think it's possible if we could just like scoop it to get it out of the rock?
So this is the ones that are like solid rock, right?
Solid. Huh.
Correct. I'm asking you.
Yeah. No, I definitely feel.
What time is it now?
I don't think it's loose rock.
No,
Oh, you see it's great.
Yeah.
19,
Yeah. That looks like solid rock.
So this scan 2 would have been taken almost 6 hours. It's all right.
thing is that the legs will break.
Break, yeah.
Go,
Yeah.
if you turn on auto snap.
I mean, maybe we can look for other one that is more suitable. But do we have time? They say that it might not be that easy to sample those without breaking. But I guess it's up of the time that we have.
It's attached to like on a rock, so I don't know if we'll be able to scrape it off without damaging it. We can try.
Yeah, when we've tried in the past to like pick it up with the nips, the legs
Yeah,
break off.
they break off, yeah.
So we're just gonna try.
You want
With
us to try with the scoop
just
and try
gently
to jolt it up?
see
Okay.
what you can get.
Okay.
Okay.
Alrighty. You in a good spot. I'll go ahead and porch out.
It happened while we were sampling. Yeah, but that happened over like 3 hours.
Yeah, definitely. It was literally right after
Looks like
this.
it. Yeah.
I might even put a timestamp in my science.
Which one are you gonna try to get?
Right before we did
Trying to
it.
look at this, yeah. That one might be free.
So
If you wanna try and grab that one.
10:45 a.m.
Okay.
Happened
Which one?
This scoop right here.
Oh, okay.
That would have been around 16:30.
So then-
16.
The Gus was on at 10:30.
So he forgot to do it. He just came off shift. I said, hey, did you fire that? He said, no, I forgot. And he ran in and he must have done it right at the turn
Yep.
at 11.
Right. So right now, Ruth, what I think you should do while you're getting set up is just go ahead and log a sample here
Okay.
for a Niskin and put a note here that this probably happened at like 1600,
Now you can always reposition.
1700, and then we need to look it up. Just so we get it logged
Grab them.
and give it a number.
Amazing.
And then do that concurrent.
Un saludito a Sammy.
And I can
Acá.
No, there's still, yeah, but for
Estamos finalizando el buceo.
here, 'cause we didn't know that there's a
Vamos a muestrear con este aparato que vemos aquí, que es como una pala, a tratar de muestrear una de estas estrellas quebradizas. Y aprovecho sí para enviarle
sample logging issues on today's diet.
un muy cariñoso saludo a Samia.
Alright, so that's all I can probably contribute.
Ya pronto nos vemos.
That's them firing it right
That's
here.
the one. There you go. Free. Yeah. Nice.
So if you write in that it was at that time.
Yeah. So the one
Thank
we're
you for
talking
looking out
about
for
is
me.
right
I'm glad we
there.
got it settled.
Yep.
Sorry, Nancy, do you want here?
Yeah, maybe we can like try it with the ones that we have around to see
So
which
let's
one
also
comes
make sure that we're
easier.
logging this.
Like just like poke it.
Did we get, before you pick it up, we need to take frame grabs.
Okay. Yeah. Okay.
Okay.
Which number is this, Roof?
This will be 260.
260. All right.
Happy with that?
Yeah. And can we get one with the lasers, please?
They're on. Yeah, I just
Okay.
had zoomed in out of the picture.
That's perfect.
Okay.
You-
I got it.
Bring it up? Yeah. Thanks. So
Alright.
260.
Alright.
All right. Let's go ahead and let's give it a try. Before, okay. Sorry. Can we do a zoom on that crustacean that just appeared there? That's really
As zoomed as I can get there, but.
All right. Okay. That was. Do you think you can annotate that, Lou? Like a screen grab of the close up of that crustacean?
Yeah.
It's not gonna be
what
a
are
sample.
you? What do you?
Just a crustacean.
Crustacean.
Unknown. Yeah.
Is that the thing on the left or?
Yeah.
Okay.
All right. Sorry to interrupt. Tyler.
You good?
All right. Let's do it. if you can get the crustacean, it'll be awesome. But no pressure.
We all had to lay down.
Yeah.
Oh, okay.
Gotta move it.
Yeah, you thought, it's like, you guys thought I was slow.
Crawl into the.
Maybe? Mm-hmm. Just invite him over.
Do a just start call for us.
First
Yeah.
challenge is getting them to crawl in. The second one is to get them to fall out.
Huh.
Yeah, I want to break it this week. See that.
Oh, there you go, buddy. Bye.
He's quick.
They're pals, huh? They're like hanging around.
Just goes whichever capsules
It seems like it. It's like he gets when you touch him from one way, he's like, "Ooh, that's where the danger's at."
Yeah.
Go the other way. Yep.
Yep.
straight to good ones.
Oh boy. Oh, that might be nice actually.
Runs off the end of the rock.
Yeah.
Towards the end, yeah.
Alright, we can control him.
Control them and catch it.
Yeah,
you think?
maybe. Might take flight. Might be able to scoop him. But it'd be hard to keep him in there.
Alright, now clear. I got plenty of space.
So I do see a comment. We are trying to avoid the slurp just because this brittle starts. Come on, let's go.
Oh,
Get
come
in
on.
there. Get in there.
Go in there.
Hold it in.
He's thinking it. Just yeah. He's like, should I go or not?
You can do it.
Yes.
It's not falling for your trap. It's a trap.
What are the odds we can get the cord out here and touch it with that and crawl in?
Well,
Like with the other?
no shoulder. It's gonna be a bit challenging.
Yeah, if we switch control, this one will drop.
I don't know. Might stay there. Problem is you can't switch it. If he goes in there and he comes out, you don't have enough time to react.
We, as I was saying, these brittle stars are really fragile. So if we try to slurp them, they might break. And having all the parts and the animal as a whole is one of the most important things in order to identify it in a proper way. So yeah, let's see. Okay, I see what you're doing.
She's scared? Now you're not scared?
Uh-huh.
There he
Oh,
goes.
oh, 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 oh. Wow, man. That's
Oh.
Yeah,
In
yeah.
my scoop? Why?
Well, you might
I think
fly up
I
if you
think
just
yeah, I
give
think
him
you
a
yeah.
little jolt there. There he goes.
Oh, not today.
He's gonna go hide.
Hey. Hey. Come
You almost got her. Oh no, she didn't like that.
No. The leg there.
Huh.
You can maybe cajole it into the sand a little bit further, then you can maybe scoop it. You know what I mean? Yeah. Like get it out into the silty area. Yeah. And then let it rest, and then try and get it scooped.
Yeah, that's a good one, man.
Just sitting on the edge of the rocks right there. Down. Well, if it climbs up in there.
I think she's
when it's
thinking it.
healed, then- Oh.
Maybe there.
Oh, there you go. That's
There you go. Okay.
And then this way, as you try and scoop, you're scooping more towards the silt anyway, so it's gonna run where you want it. Yeah. If you try scooping the other way, it may run back to the rocks. Another shot here. There you go. Now it's lining up.
Pretty fast, man.
Yeah.
Wow.
They're gonna run for the rocks.
There you go, there you
Oh.
go, there you go, there you go. There you go, there you go, there you go, there you go, there you go. Ah.
So close.
Back to the rocks. Okay.
Second sample purple?
They said they don't want to use the suction sampler because they don't want to break the leg.
Yeah, if you feel it could be done, that would be great
Well, that was
That was
because
our first suggestion.
You just don't put maximum suction in
Okay.
as soon as it comes in.
let's give it a try, and yeah, that will be our last shot because yeah, the chief says that just have 3 more minutes, so let's do it with the.
Clearly, and outrun time. The wiggler versus Ziggy.
The Wiggler wasn't losing last night, was he, sir?
Well, I don't know.
Oh.
Well, Luigi won at the very end, so I don't know.
No, you were evading the challenge, I think.
That was sweet.
That's all. That's another word for it.
I'll be back in a little bit, Doug.
you're gonna like slurp it all the way
We'll have
Into the
to
jar?
into the jar.
into the jar. Okay,
Yeah, is
that's
that right?
yeah. Just
We are on jar 5 right
Yeah.
now.
Are you ready?
jar 5. Okay.
Good.
It's easy.
All right.
Not lined up with the jar. Yeah.
There you go. There you go.
Do you want some of the sediment as well, Sergio?
No. No, we're good. Yeah.
Just keeping an eye on number 5.
Hopefully it didn't break. Just. Things getting there. Mhm.
Come on, buddy.
Long way to go.
Yeah. Yeah, especially if you're on over in this section.
Yeah. She'll get there. She's definitely a fighter.
Yeah. Trying to get a little sediment in there so we can see how that flows through.
Okay.
Alright, so a bit of dirt coming. There's the dirt.
Alright, yeah, maybe she's attached to the hose.
One suction.
So I got it full now.
Alright. In any case, I mean, we definitely slurp. Okay, I think I see something there. It's just like stuck like this.
Oh.
That was small too, so that's
What happens if we come up that way and it's like with the arm looking up or looking down to avoid any escape?
I mean, whenever we come all the way up, the arm's gonna turn
Okay.
down a little bit.
Gotcha.
Once we turn hydraulics off.
Okay.
So
Try
it's
taking some more dirt. I'll put it through.
Yeah. Yeah, the thing is that the
dirt flush.
the chief is telling me that we have run out of time. So yeah, maybe one more slurp and off we go.
Yeah. So maybe hang inside of the hose for sure.
Yeah. I mean, if at the end she escapes, well deserved.
Alright, Tyler, you can probably steal that hose. I'll just keep an eye on it. Here comes the dirt.
Yeah. All right. So that was 260, right, Ru?
260, yes.
And gonna put a question mark.
Yeah, I'll put a question mark too.
Keep going until that clears up. Not saying that it's coming through, but I'll do it till it clears up. Just
because we are over time.
Okay.
So yeah, I appreciate it.
Yeah, yeah.
Just gotta.
I'm gonna go ahead and stop the pump.
Okay.
And then I'll just index to the next jar. That way, in case it is in 5, it's in 5.
Okay. Nice.
All right.
Thank you guys.
Yep, no problem.
That was really exciting sampling. Yeah. Really nice kills, man, with the poking and yeah, that was pretty impressive.
Yeah, it's more fun than slurping, but
All right, so we're about to end our dive. We are going to start ascending really soon. So I invite you to stay tuned to this live stream because usually we see many fascinating creatures up in the water column.
Yep. You ready for us to come off bottom?
Yes, ready to come off bottom. Thank you.
Bridge ROV.
Vamos a iniciar nuestro ascenso, lo cual quiere decir que ya nuestro tiempo de fondo ha terminado. Prácticamente ya quiere decir que pues todo el muestreo pues finaliza. Sin embargo, pues durante el ascenso en la columna de agua también hay interés particular en algunos organismos, así que pues vamos a mantenernos aquí atentos y les invito también a que se queden atentos y atentas a look at that one, that was
What's that?
no, just that there was another brittle star like really
course.
just laying
We're
there.
not looking for them.
Exactly.
We're not looking for them.
Manténgase sintonizados en sus pantallas porque vamos a iniciar el ascenso y definitivamente que, bueno, al menos a nivel personal, y hablo por mí, muchos de los animales más fascinantes y más interesantes se encuentran en la columna de agua, así que atentos y atentas. Y bueno, con esto me despido. Con esto me despido. Ha sido un placer para mí estarlos acompañando en esta última porción del buceo. Mi nombre es Sergio Cambronero y bueno, invitarles a que se sigan sintonizando estos buceos. Tenemos, si no me equivoco, 2 buceos más durante los próximos días, así que pues aún queda muchísimo que explorar, muchísimo que ver. Y bueno, pues si tienen alguna pregunta, algún comentario, lo dejan en los canales de YouTube y Facebook, y ya sea ahorita o en los próximos buceos les estaremos respondiendo. Muchas gracias y pura vida.
Thanks, everyone.
Thank you, Sophie. Thanks, everyone. Stay tuned for midwater creatures. Woohoo! Brittle star 1, Wiggler 0.
it might be the jar.
Oh, yeah, that's right. Yeah.
Okay, and now it's Ascent time. Thank you all so much for watching today. I hope you enjoy all of the creatures. We'll still be around if you have any questions, and we will be back later tonight. We are going back to Dorado Outcrop for dive 630. We will be continuing some of our experiments there and retrieving samples. And yeah, we will see you later tonight. Thanks so much for joining us, and we'll see you later.
Spot critters.